Bob Lowry, CEO and founder of Public Labs, and I want to welcome you to our webinar today on streamlining high-throughput screening and lead discovery with the Transcreener ADP2 Kinase Assay. Meera Kumar, our Senior Application Specialist, was initially scheduled to present this webinar, and she's in the middle of a big screen. So I'm standing in for her, and I hope that I'll be able to do a good job on this. And I always enjoy having the opportunity to speak directly to our customers about my favorite subject. So let's get moving, and we have a lot to cover, so we'll just jump right in. So today I want to cover, first of all, some background on the Transcreener ADP assay and some of the competitive advantages we have over other assay methods, and with the goal, of course, being sensitive detection of kinase initial velocity, because I know that's what you're interested in. Using any substrate in any ATP concentration. We'll cover some specifics, the compatibility with 1536. Really the outstanding reagent and signal stability, and how the assay gives you a lot of flexibility in terms of screening formats and automated liquid handling. And then we're going to look at some comparison things between Transcreener and one of our major competitors out there, ADP Glow, for detection of kinases, and we'll look at some pilot screening data. And then show you how some of the differences in the assays in terms of our sensitivity enables us to obtain accurate IC50 values, even for very potent kinase inhibitors. And last, I'll show a few slides on using the assay in kinetic mode, which is really a nice thing to be able to do, because it enables you to get things like drug residence time, which are really becoming a key factor now in lead optimization. So the Transcreener assays, some of you may be aware, are basically immunodetection assays for nucleotides. And there are 4 basic assays which we've been converted into different formats using different readouts, but they all detect a nucleotide, which is the product of a number of different enzyme reactions. And that's really the power of the platform. With these 4 different assays for AMP, GMP, ADP, GDP, and UDP, we're able to detect literally thousands of different enzymes that produce these nucleotide products. And this is a schematic for the ADP assay, and all the Transcreener assays work similarly. So you see a kinase reaction is producing ADP, and that competes a fluorescent tracer off an antibody. And then the free tracer has a lower polarization value, and so the signal is a decrease in polarization. We typically graph that as the net change in polarization, so that the graph is a positive change. The other assays, the basic mechanism is essentially identical. We just have different formats in terms of what we conjugated to the antibody. So for fluorescence intensity detection, we have a quencher that's attached to the antibody, so that the tracer then is quenched when it's bound, when it's displaced by ADP. Then you get an increase in fluorescence. With the TR-FRET assay, we have a lanthanide chelate that's attached to antibody. So we have a FRET that occurs between the chelate and the fluor on the tracer. When that's displaced by ADP, then you have a decrease in the FRET value. So the assays all work essentially the same. They're competitive immunoassays. And the thing to note here is we're directly detecting the formation of ADP. So there's no intermediates in between the binding of the ADP to the antibody and the assay signal, no kind of coupling reactions or anything like that. And the assays rely, of course, then on antibodies which are able to selectively recognize, in this case, ADP, and not bind with any significant degree of cross-reactivity to ATP. And that's just shown graphically here. And so if we do competitive binding experiments, we're looking at ADP here on the left. ATP then displaces the tracer with about 140-fold less competency So we found that in order to generate good assays for initial velocity enzyme detection, we need about 100-fold selectivity for the product nucleotide versus the substrate nucleotide. So what that enables you to do then is to directly detect initial velocity enzyme activity. So here we're looking at protein kinase A, and we're doing enzyme titrations here. And then what we do is we convert the polarization values to ADP production using a standard curve. So a standard curve for ATP conversion, we start with the initial concentration of ATP, in this case 10 micromolar, and then we add ADP and remove ATP, decrease ATP proportionately. So it essentially is mimicking an enzyme progress curve, the conversion of ATP to ADP. And using that standard curve, then we're able to convert the polarization values directly to ADP amounts. And you can see that then when you graph these enzyme reactions over time, we're looking at linear formation of ADP. And of course, that's where you want to be, in that initial velocity region where you haven't consumed a lot of your substrate yet. So your reactions are still linear. So we're able to, by adjusting the concentration of the antibody, we're able to tune the dynamic range of the assay. So at a given antibody concentration, the assay has a dynamic range of about 1.5 to 2 logs, because it's a binding assay. So you're going to get a binding isotherm, and that's about 1.5 to 2 logs. But by changing the antibody concentration, we're able to, as you see, as we increase the antibody concentration here, we're right shifting the binding curves so that we're able to detect different ranges of ADP concentration. And there's actually a linear relationship then between the antibody concentration, the optimal antibody concentration, and the starting ATP concentration, and that's provided in the protocol. So in some cases, it's not even necessary to do the antibody optimization yourself. You can take it right off this linear relationship. And we found that, yeah, the EC85, that is the concentration of antibody that gives you about an 85% shift of the maximal shift in the fluorescence polarization value of the tracer, is the best concentration of antibody to use for initial velocity detection. So that's how the assays are set up. And we're going to use that concept of the standard curve again and again. So here it is again here, shown where we're looking at the percent conversion of ATP to ADP, starting at different initial ATP concentrations. So this would be, if you want to run a kinase, typically if you're running kinase for screening or for inhibitor profiling, you often want to use the Km concentration of ATP. So you need to be able to have a flexibility in terms of your ATP concentration. And you can see by adjusting the antibody concentration here, we're able to generate essentially identical response curves for the assay using all these different initial ATP concentrations, from as low as 100 nanomolar all the way up to 1 millimolar. So the assay is quite flexible with respect to the concentrations of ATP that you can use. And of course, because we're detecting ADP and not the phosphorylated product, it's a universal assay with respect to the type of substrate, acceptor substrate that you can use. So here we're using a number of different protein kinases with either peptide substrates or full-length protein substrates. And the assay works equally well, independently of what the acceptor substrate is for protein kinases, and also for other types of kinases. So for lipid kinase and metabolic kinases or carbohydrate kinases, works equally well because we're detecting ADP, of course, and not the phosphorylated product. So it provides a lot of flexibility there in terms of the types of acceptor substrates, which, you know, if you're running, for instance, a kinase program and you want to compare different peptide substrates, say, or different protein substrates to see what your differences in activity are, you know, you're going to be comparing apples to apples. So it's very handy in that way. So another property of Transcreener that I think is unique to this assay is the stability. And here we're looking in particular at the FP assay. The TR-FRET assay has similar stability as does the FI, but the FP assay is really the most stable. So the reagents on the left, you can see are essentially stable for, well, certainly as long as you'd ever probably leave them laying around the lab. But in practical terms, you know, if you're doing a large high volume screen, you know, on an automated platform, you know, there's a good chance that your reagents are going to sit on the deck for a while, you know, before they're dispensed into plates. And you don't have to worry about any decay in the reagents in terms of their performance while they sit on the deck while you're waiting. So that, I mean, that's a very, gives you a lot of flexibility as far as your screening protocols that you run. Again, then when we're looking at the signal, so this is after, you know, quenching the reactions. Again, we're just looking at standard curves here. And you can see that the signal is quite stable as well. So if you run a lot of, if you're running a lot of plates and you can't do reads right away, and the plates are sitting on the deck for a while before they're read, it's not an issue. And all of the assays have at least overnight stability, and you can see that the FP assay, it actually has stability out beyond 48 hours. So both the reagents and the signal are quite stable, which gives you a lot of flexibility as far as especially running large screening protocols. So all the data I've been showing you is in 384 well plates. You know, our final volumes after addition of detection reagents are 20 microliters. The assay can certainly be used at 1536 as well. And in fact, most of our customers who run large screens use 1536. And again, these are just showing you standard curves here for conversion of 10 micromolar ATP or 100 nanomolar ATP to ADP, and looking at both the change in polarization in red and then the Z prime in blue. So you can see by the time you've gotten to 10% conversion, you've got a very good change in polarization over 100 millip, and the Z prime is, well, in this case, it's almost 0.8. So even in 1536, you know, there's no problem running the assays in that format in a smaller volume. At the lower ATP concentration, and this would be quite a low concentration used for a kinase assay, you can see that you need to go a little higher ATP conversion in order to get that really good Z prime value. But it's still within the initial velocity region. So the assay is quite compatible with 1536. So one of the criticisms that is sometimes leveled at antibody-based assays is that you know, there's going to be potential differences, you know, because of lot reproducibility. And I've never really understood the origin of this criticism, because of course monoclonal antibodies are highly reproducible from lot to lot. I mean, as all of you are aware, there are many therapeutics that are comprised of monoclonal antibodies, and they have to be reproducible from lot to lot. And similarly, we're able to get very good reproducibility lot to lot in all of our antibodies. So this is again showing you standard curves here at 3 different initial ATP concentrations, and the 3 colors in the standard curves are different lots of antibody that were produced within a period of about a year. And you can see that the plots are superimposable. So we don't have any problem with the reproducibility of our antibody and getting similar responses to lot to lot. So if you buy a batch and then you do a screen, and then 6 months later you want to do some inhibitor profiling and it's a different batch of antibody, you're going to get the same response. So also very important, this is some work that was done early on by some investigators at Eli Lilly who were adapting the assay there. And they were able to show, they wanted to validate that the inhibitor potencies that they observed with the assay would be similar to what they were observing in their gold standard radio assay. So that's the FB stands for filter binding. So that's a radio assay, and then the transcreener data is on the x-axis. And these are IC50 values with protein kinase A, and you can see that there's very good concordance between the transcreener assay and the gold standard radio assay. And a number of companies have since done these kinds of experiments as part of their internal validation for the assay, and this is typically what is observed. So there's the potencies that you measure with the assay are accurate. Another thing in terms of concordance, so we have three different assay formats, as I mentioned, detection formats. There's the FP, the TR fret, and the fluorescence intensity. And sometimes, investigators will use one for a high-throughput screen, and they'll use another one for hit confirmation. And this is just showing that, yes, you get very good concordance between the inhibitor potencies measured with the three different detection formats. In this case, we're looking at a number of different kinases and different inhibitors here, not just protein kinase A. And just one additional thing to add here in terms of the general information is that, we've validated this assay, and we do the validation either here or sometimes the instrument vendor does it in their own labs. So validated for specific performance specifications, and those are Z prime of greater than 0.7 at 10% conversion of 10 micromolar ATP to ADP. And we provide on our website, the exact filters and instrument settings to use to get this optimal performance on all of the major multi-mode readers out there. So I want to focus for a little bit now on the differences, specifically between Transcreener and the other ADP detection methods out there. And the main point I want to make is that, as I mentioned before, Transcreener is direct detection of ADP. ADP is binding to an antibody, displacing a tracer, and you're getting a change in the fluorescence properties of the tracer. The other ADP detection methods aren't actually detecting ADP. They convert ADP to something else through a series of enzyme-coupled reactions, which is then detected. So our assay is simpler, and we believe for that reason less subject to interference. So in these slides, which I'll put them together quickly, this is the mechanism of luciferase-based ADP detection, as in Promega's ADP Glow assay. So it involves one pre-treatment enzyme, which eliminates the residual ATP, that's an adenyl cyclase, and then two post-treatment enzymes, which is a creatine kinase, and then the luciferase, which is actually results in the detection of the light-based signal. So it's a two-step detection where you first, if you're familiar with it, where you first add these pre-treatment enzymes and then you add the detection mix. But again, it involves three enzymes that are involved in actually generating a signal. This is another resirufin-based detection method, fluorescence. This was DiscoverX's ADP Quest assay. And you can see also in this case there are three enzymes involved that are converting the ADP to something that's, in this case, the resirufin, the fluorescent resirufin that's actually detected. So again, a fairly complex reaction mechanism compared to the simple immunodetection with Transcreener. So I want to do some direct comparison here now with Transcreener and ADP Glow. And the first thing I'm showing here is just this 10 micromolar standard curve. And I've showed you a number of these. So we start with a given concentration of ATP that's depleted, and ADP is added proportionally. We recommend to our customers that they plot this as a semi-log plot. Promega recommends that they plot it linearly, so we're just doing this according to the manufacturer's directions here. And you can see that for both assays, these standard curves were done with an N of 16, so we're able to generate Z prime values. And you can see that for both assays, by the time we get to 10% conversion, that is 1 micromolar ADP, you've reached about your maximum Z prime, so that's good. The Z prime that we observe with Transcreener is a little bit higher, but I mean you're up above 0.7 here, so these are very good Z prime values for both assays. So if we look at enzyme titrations then, and I'll mention up front that because of the 2 reagent addition with the ADP GLOW assay, their protocol is a little different. So we recommend our customers run a 10 microliter enzyme reaction and then add 10 microliters of the detection reagent. It's a single addition. With ADP GLOW, they recommend a 5 microliter enzyme reaction, and then you add 5 microliters of the pre-treatment that removes the residual ATP, and then 10 microliters of their detection mix. So they're running a 5 microliter enzyme reaction. So we did this comparing our standard format as well as running a 5 microliter enzyme reaction with a Transcreener assay, and then we had 15 microliters of detection. So the end volume is the same in all of these. We just wanted to see if there was any difference. And the other difference is that we recommend using what we call the EC80 concentration of enzyme. That is the concentration of enzyme that gives you about 80% of your maximum signal, and that's indicated here by the dashed lines. Because we know that A, you're getting a very good signal. You can see that it's almost 200 millip, and B, at that enzyme concentration, you're getting somewhere around 10% conversion, which is where you want to be in terms of your initial velocity. In contrast, ADP GLOW recommends using between 5 and 20 times the signal background. Okay, and signal background ratios aren't really relevant for an MP for a polarization-based assay, so we don't use that parameter. And so I've marked both those for their assay. This is the EC80 concentration, which is where we would recommend. This is at the 10 times the signal background, which is right in the middle of what they recommend. And then I'm looking at enzyme usage here in the table. So with our 10 microliter reactions at our EC80 concentration, we're using about 8 femtomoles of enzyme, about 0.8 nanomolar. If we cut that down to 5 microliters, it's about 5 femtomoles. With the ADP GLOW assay, if you use the EC80 concentration, it's quite a bit more enzyme. If you use their recommended concentration at 10 times signal background, you're at 15 femtomoles. So it's still, about 3 times as much enzyme as the Transcreener assay in the same volume. And why does that make a difference? Well, first of all, if you're doing a large screen, if you can cut down on enzyme usage by even 2 or 3-fold, it can make a big difference in cost. But secondly, and maybe more important, is that if you're looking at high potency inhibitors, which hopefully at some point you are, that is inhibitors with low nanomolar IC50s, in order to avoid depletion of your inhibitor with your enzyme, you have to be using an amount of enzyme that's, down in the same, hopefully lower than your inhibitor concentration, certainly not significantly higher than it. Otherwise, you're actually titrating out your inhibitor with your enzyme. And so this is some data from a project that we ran for a customer where they wanted to screen these various enzymes. They wanted to use these enzymes for profiling inhibitors, inhibitor potency, and their goal was they wanted to be at 5 nanomolar or below with the concentration of all their enzymes for this inhibitor profiling project. So we were able to achieve that for them quite easily with the Transcreener assay. So being able to use those lower quantities of enzyme, that really can serve as a practical advantage, both in a screening and later in a lead-op or selectivity profiling mode. So we also ran pilot screens So this is a mock screen. So this is not actually a screen with enzymes. So the wells contain 9 micromolar ATP, 1 micromolar ADP. So it's mimicking, a kinase reaction, a completed kinase reaction. And we're screening about 10,000 compounds from a Cambridge diverse set. This is a very good high quality library, lead-like compounds. We've actually used it for a couple of screens internally, and we end up with it's been filtered for things like panes and reactive compounds and all that. The reaction conditions, we're using the manufacturers recommended buffers, which are actually very similar. We use we use Bridge, they use BSA, but other than that, the reaction buffers are pretty similar. And again, the protocols are somewhat different. We set up a 10 microliter mock reaction plus or minus compound, add 10 microliter detection mix. They have the 5-5-10 format. And these are the results of the pilot screens. We ran this with the Transcreener ADP assay, the TR-FRET assay, and then an ADP Glow kit. And the first thing that you'll see is that, so for our assay, the negative controls, these blues, are the 10% conversion. The positive controls here represent 0% conversion. So this would be complete inhibition. Same for TR-FRET. Okay, theirs is reversed, so that the negative controls are up here. This is 10% conversion. This is 0% conversion. So, and in either assay, hits would be showing up in between those two. And then the horizontal lines here represent 3 standard deviations, which is generally used as an indication of a hit. It falls someplace outside that. So the first thing to notice is that there's very few interfering compounds with any of these assays. And there are fewer with the FP assay than with the TR-FRET. So here's some compounds here that would be interfering compounds. Here's a couple here out of this entire 10,000 compound set. And that's consistent with what we see and what we tell our customers. Customers doing a large screen, we recommend they use the Transcreener FP assay. Despite the reputation that TR-FRET has for lower levels of interference because of the gated emission and the delayed detection, we find, and honestly, our customers through the years have found that the Transcreener assay, the FP assay, gives the best results in terms of compound interference. Now, you might be able to find libraries where you can get a higher level interference, but in general, that's what we found. So with ADP Glow, again, very few compounds that would show up as false positives. There's, I don't know how many there are, 2 or 3 or something like that. There's a little bit more scatter, both in the positive control data and in the actual screening data. And then there are a number of compounds here which show up as I guess they would be enzyme activators, which is a little bit odd. And I don't know exactly what that means, but these are typically not compounds that you would be interested in if you were doing a screen. So if you look at the Z and Z prime values, so the Z being the combination of dynamic range and signal window between the controls, and then the Z prime being for the actual compound data. The FP and the TR-FRET, this is what we typically see. Z and Z prime values of about 0.7. They were a little bit lower with ADP Glow, but still within an acceptable range, and probably mostly because of this the scatter that you see here. This is actually the first time we've compared the three assays, so you're seeing some results here hot off the press. Last thing I want to focus on, drug target residence time. So this is something that, I'll make this quick. We're just about out of time here. That you're able to do with the Transcreener assay because the ability to use the assay in a continuous or kinetic mode, that is to add the detection reagents directly to a kinase reaction and then read the signal at intervals to follow the enzyme reaction over time. So drug target residence time is basically the time that inhibitor spends bound to a target before it dissociates. And that's increasingly something that people are building into their lead optimization efforts. You know, I was just at the drug discovery chemistry meeting in San Diego, and residence time has become, over the past really just 2 or 3 years, something from, you know, this kind of new idea, this concept that was put out there, to something that is now a pretty typical part of a kinase lead optimization program. So we were glad to see that. And so the ways to measure it are either biophysical methods, things like surface plasmon resonance, or to look at ligand displacement assays, or, as I'll show you, to do what's called a jump dilution method. So in this approach, and we like this method because you're actually looking at the dynamic catalysis. So you're looking at the displacement of the inhibitor during dynamic catalysis of the enzyme. So you combine an inhibitor with enzyme at a saturating concentration. So essentially, all of your enzyme is bound with inhibitor. You're 10-fold over the IC50 value. And then you dilute that 100-fold so that the inhibitor is no longer saturating. So now you're only at 1/10 of the IC50 value. And then you monitor activity. And what you see, and it's easier to illustrate in the lower right here, these are the actual raw polarization values, is that you know, inhibitors that bind very loosely, the activity will recover very quickly, whereas inhibitors that are bound more tightly, the activity recovers slowly. So you're looking at the recovery of activity as the inhibitor dissociates. So a tight binding or long residence time inhibitor is going to dissociate and the activity more slowly, and the activity is going to recover more slowly. And then using you know, this first order rate equation, you're actually able to then determine the tau, which is 1 over the dissociation rate. So determine the inhibitor residence time. And the advantage of one of the advantages of doing these in this jump dilution approach is, you know, we're doing this as a multi-well format. So you can do a lot of these reactions. You can look at a lot of different inhibitor residence times on a single plate. So it's quite powerful that way, rather than running separate SPR reactions, or even if you're using a multi-channel SPR instrument. There's a lot more involved in terms of optimizing, you know, the binding of the attachment of the enzyme to the chip and all that kind of thing. This is all in solution, and we're looking at dynamic catalysis. So, and this is from a paper we published just last year. It seems like longer. And so looking at and comparing the residence times that we determined for a number of inhibitors with different kinases, and comparing those to some of the literature data for some of the biophysical methods and ligand displacement assays. And although the rank order of things tended to be similar, you know, some of the values that we got were very different. And you know, if I'm a biochemist, I am a biochemist, and I'm looking at data and I'm saying, well, do I want to, you know, what do I want to believe? You know, the displacement of a inhibitor from an enzyme that's bound to a surface, you know, where it's conformationally constrained, or alternatively looking at the dissociation during catalysis. You know, I would pick the latter. So we're kind of lobbying for this jump dilution approach as a more really physiologically relevant measure of drug target residence time. So just a few minutes over here, I want to thank everyone for their time. I know you all have plenty of work to do, and I appreciate you taking the time to spend with us in this webinar. And I want to thank Mira, who did a lot of the work that was presented and certainly was involved with designing the experiment, and Josh Sether, who helped her, especially on these the pilot screens. Thank you all very much for your time.